So today we're going to take trips 2 and 3 through the digestive system, and then we'll save digestion and absorption, the final trip, for our next lecture. I think we'll get through these two today. We'll have to see. We're also going to be filling out kind of a summary sheet as we go, as for important things. And then things that won't really fit on that summary sheet, I'm going to highlight in yellow for you to remember. Okay, so we're going to start and go top to bottom with motility. Then we'll go top to bottom with secretions, and we'll be filling out that summary chart as we go along. Okay, so motility is propelled through mixing and propelling of food using the smooth muscle of the GI tract. We'll start with the mouth. The mouth is going to chew food and break it mechanically into smaller pieces. It does mix food with saliva. Chemical digestion begins here, but doesn't really occur until we get to the small intestine. So for the mouth, we're going to chew food into smaller pieces, form that food into a bolus using saliva, and then using the swallowing reflex, which we'll get to in a few slides, then that will propel that food bolus through the laryngopharynx and then down to the esophagus, where it will keep going until it reaches the stomach. The esophagus, the main function is peristalsis. Primary is that swallowing reflex, and secondary is to continue the movement of that bolus. There are a few structures that are involved with the swallowing reflex. So those structures are going to include the uvula, because that is going to close off the nasal cavity, and you don't want to get food up in the nasal cavity. That mucosa does not appreciate it. So when food gets chewed up, the tongue will use the hard palate to form a bolus along with the saliva that's been produced by the salivary glands, and then it will push that food back. It causes the swallowing reflex, which is the uvula, moves up to close off the nasal cavity. And then the upper esophageal sphincter, sometimes called the laryngoesophageal sphincter, is going to open. Normally, the esophagus is closed. Normally, the sphincter is closed. So when it's in a resting state, it's closed to prevent air from moving down the esophagus. So as the food bolus goes into the laryngopharynx, the epiglottis is going to close off the trachea, and that upper esophageal sphincter will relax, opening the esophagus and allowing food to move through. Once food has passed through the upper esophageal sphincter, the epiglottis is going to stay in that upright position, reopening the trachea. The uvula moves back down, reopening the nasal passageways so that we can breathe either through our nose or our mouth, and it will make its way down the respiratory tract. So the structures that are involved are the uvula, the epiglottis, the upper esophageal sphincter, and then the lower esophageal sphincter, which is also called the gastroesophageal sphincter. Once the food bolus enters the esophagus, then the circular muscles contract and push that food bolus through peristalsis down past this lower esophageal sphincter, which is also at rest, normally closed. So both of the esophageal sphincters at rest are closed. The upper to prevent air from entering, and then this lower esophageal sphincter, also known as the gastroesophageal sphincter, prevents the gastric juices from going up into the esophagus and burning the lining. So that's heartburn, but it would damage the esophageal lining. So both of these sphincters, unless the esophagus is being utilized, are going to be closed. So you do need to know the structures, the uvula, the epiglottis, the upper esophageal sphincter, and the lower esophageal sphincter and their functions. Swallowing contains both a voluntary component to the reflex and an involuntary. For the voluntary part, the tongue is going to push the bolus into the oropharynx. So once we are finished masticating it and mixing it with saliva, then it gets formed into that bolus and moved into the oropharynx. That initiates the involuntary part, where the upper esophageal sphincter opens, the uvula moves up, the glottis moves down, and that lower esophageal sphincter is also in the stomach muscle is going to start to relax. There is a secondary movement if the first is not complete. We will continue with that swallowing reflex until the bolus moves past that upper esophageal sphincter. Okay, so actually we're going to go ahead and move over to our summary sheet, calling it my in-class summary. So for the mouth, for motility, we have chewing and forming the bolus. For the pharynx and the esophagus, we have the swallowing reflex. And for that, you need to know the structures, the uvula. It's going to close off the nasal passages. The epiglottis closes off the tracheal passage. And then we've got the two sphincters. Pharyngeoesophageal, which is the lower sphincter. I should just count on an auto spell check. And then the lower one is the gastro. And then the other movement that occurs in the esophagus is peristalsis. All right. So now we're going to move on to stomach motility. The stomach has three purposes. It's going to store food via receptive relaxation. So if you think about it, blood vessels are also made of smooth muscle. And if we have a blood vessel and we increase blood pressure and it expands, normally that will trigger a constriction, a contraction to return to normal. So the stomach is a bit unique in that it's also made of smooth muscle, but when it stretches, it relaxes instead of contracting. So that is pretty unusual and unique to the stomach. So as the stomach fills, those rugae are going to relax and allow for the stomach to fill with contents. The second purpose is mixing of the food with stomach juices. So this is the churning, and this is accomplished because it has the circular layer, it has the longitudinal layer, and then it has a third layer of muscle that is an oblique layer. So that oblique layer is going to enhance the churning action. And then emptying into the duodenum, and we'll look at what controls that rate. So we mentioned receptive relaxation to store food up to about 1 liter. Once you go past that point, you may end up vomiting it back out because you've exceeded the storage capacity of the stomach. There are 3 phases of digestion. We'll probably go back over these when we get to digestion and absorption. It kind of fits a little bit better there. But there are 3 phases, and I'm actually going to go to this one. Digestion occurs, it starts with the cephalic phase of gastric motility. When we see food, think of food, smell food, are driving over to a restaurant, the autonomic nervous system via the vagus nerve is going to start the process of peristalsis, and it will start salivary amylase to saliva being released. So kind of the thought of food starts this gastric motility, then the gastric phase, and then the intestinal phase. So the gastric phase is meant to enhance stomach motility. So gastrin, we're going to be increasing hydrochloric acid, we're going to be increasing pepsin release, and it turns into pepsinogen, increase of gastric juices. So in gastric phase, we're getting ready to increase all of that stomach action. And then the intestinal phase, the duodenum is going to say, hey, wait, things are going a little fast here. We need to slow things down. So we'll review this when we get to secretions, but this duodenum portion of the small intestine, when it gets distended, will end up, there's a hormone called secretin, and it's going to inhibit the gastric activities. And then if there is a high fat content in the chyme, cholecystokinin will be released to slow down motility because it takes longer to digest fat. So you can see that these are highlighted. So you do need to know the 3 phases and be able to match them to the events that are occurring. So for movement, gastric peristalsis is going to churn and mix the food and deliver chyme, which is like a liquefied food. It's food plus gastric juices to the duodenum. And there are about 3 to 5 contractions per minute. And because of the way the stomach is arranged, those contractions are weaker in the body and stronger towards the pylorus. The pylorus is that section down by the pyloric sphincter. The rate of chyme delivery into the duodenum is going to be controlled partly by food volume. So the more food, the more intense the contraction and the more chyme that is released, because your stomach is saying, oh my gosh, I have all this food coming in. I need to start getting rid of it. So increase the volume, increase the chyme into the duodenum. So there are the 2 factors that determine emptying rate is food volume. So the greater the food volume, the faster the emptying rate. So the more food, the more the intense the contractions, the faster it will empty into the duodenum. And then on the flip side, the duodenum through hormones and nervous reflexes are going to slow the emptying rate. If the meal is fatty, it will slow down to allow bile salts to emulsify that fat. If it is too acidic, then it will also slow the rate of emptying to allow the pancreas to secrete more bicarbonate. We'll talk about reflexes in a little bit. Okay, so the two factors are food volume and then the composition of the chyme. Is it fatty or is it acidic? Okay, so we're going to go back real quick to the stomach. The stomach has three functions. Okay, so it is going to be responsible for storage, and that is using the mechanism of receptive relaxation. There is churning, and that is due to the three layers of muscle. So we've got that additional one that is oblique. The three phases of gastric motility. So be able to match those. I'm not going to write out all three of them. And motility is regulated by volume, also fluidity. Fluids actually pass very quickly through the stomach. So if a meal is very like an iced cappuccino maybe, then that will pass very quickly through because it's already in a liquid form. So increase volume, increase fluidity, and that will increase, it's going to increase motility. And then you have the effects at the duodenum. So I'll just do SI composition. If it's acidic or if it's fatty, we're going to have a decrease in motility. So I have the small intestine, we'll have the large intestine, and then we'll have the contributions of the pancreas and the gallbladder. For the small intestine, since we're here, we're going to just go ahead and add that its main motility is segmentation, and that is going to be at about 9 to 12 movements per minute. There is also peristalsis, but this is housekeeping in the fasted state. And then we also will need to talk about the gastroileal reflex. So for motility of the small intestine, the main movement is segmentation, where the chyme gets mixed with enzymes. And these enzymes are in the small intestine, but they are not produced by the small intestine. The majority of the enzymes are going to be produced by the pancreas. We'll also have bile, which is made up of things like bile salts and cholesterol and lecithin. Those are going to be made by the liver, stored by the gallbladder. The small intestine will provide some intestinal juices, some mucus as well, but most of the enzymes are going to come from the pancreas. For segmentation, it's 9 to 12 movements per minute. It moves chyme through the intestine, but slowly. The other movement that occurs in the small intestine is peristalsis. But peristalsis is going to be predominant when one is in the fasting state. So segmentation is with food because it breaks it apart and it mixes it with other segments and it mixes it with digestive enzymes. So segmentation occurs with food. Peristalsis occurs during the fasting state, just to kind of make sure that there isn't anything left in that section of the small intestine. Because we want to get rid of, so food is going to stay about 3 to 5 hours in the small intestine. And that is moved by segmentation. Then when we get fasting, then these large waves of peristalsis are going to just kind of make sure that everything is running clear. The gastroileoreflex. So at the end of the ileum is the sphincter that connects it to the cecum, the beginning of the ascending colon. And it will relax with an increase in stomach motility, which is occurred by gastrin. So this gastroileoreflex means that the ileocecal sphincter will relax when the stomach is active. And that aids in removal of chyme, which is about when it enters the large intestine. Now it's called feces. Okay, so the terms change. We have a bolus in the esophagus, and we have chyme in the stomach and small intestine, and then we have feces when we get to the large intestine. The large intestine, what cannot be or is not absorbed, enters the large intestine, about 1/2 liter a day. There's still going to be a substantial amount of water that is reabsorbed. So for every 500 mils of 1/2 liter that's entering the large intestine, about 150 mils of feces are excreted. The rest is going to be water and reabsorbed. There are 2 movements that occur in the large intestine. One is called haustral movement. So this is haustra to haustra, and it's slow, 1 segment every 30 minutes. And then we're going to have something called mass movements. So a mass movement is when the whole ascending colon is going to try and clear that segment, or the whole transverse colon is going to try and clear out that segment. The descending will clear out that segment. And these occur 3 to 4 times per day. Okay, so we're going to go back real quick and add that in. So for the large intestine, we have 2 types of movement. We have haustral contractions, and these are 1 every 30 minutes. Okay. So slow, try and help with that reabsorption. Then we have mass movement, and these are of entire segments. So these are large, that's why it's called a mass movement. And then these are going to be 3 to 4 times per day. And then this is going to involve something called the gastrocolic reflex. This slide shows the reflexes that are involved with the enteric nervous system to help control motility and digestion. We have receptive relaxation, That is this first one over here, where as food fills, the stomach actually relaxes. Then we have the enterogastric or gastroenteric reflex. As the stomach fills, motility along the segmentation and peristalsis, that rate is going to increase along the small intestine. The gastroileal reflex is under control of the hormone gastrin. When the stomach starts to fill, there is a relaxing of the ileocecal sphincter. So as the stomach starts to become more active, the movement along the small intestine increases. That's gastroenteric. Then the gastroileal is going to open the sphincter to allow food to flow into the large intestine. The gastrocolic, so sometimes the large intestine is called the colon. It is going to increase the movement along the colon. So be able to match, know the concept of receptive relaxation, and then be able to match each of these three reflexes with its function. So the enterogastric reflex is going to increase motility. So food starts to move faster through the small intestine. Gastroileal is the relaxation of that ileocecal sphincter. Gastrocolic means that as the stomach becomes more active, it speeds up movement along the colon. So they will somewhat, match the functions. So this gastro part means that the stomach activity, motility, is increasing. And then it is going to have an effect either on opening up that sphincter, the ileocecal sphincter, increasing the colon movement, or enteric. You just have to kind of think of that. There's that huge enteric nervous system is going to increase the small intestine motility. To be able to match those three reflexes to their purposes, I guess. So now we're going to go back through again, starting at the top and moving through. I think we're doing okay on time. Any questions about motility before we move on to secretion?
Can you go back to the last slide, please?
Oops, wait. Sorry, my volume's down too low. Can you repeat that?
Oh, I was asking if you could please go back to the last slide.
This one.
Yes, thank you so much.
Yes. There's three reflexes. Gastro means increase in stomach motility. Then it has an effect either on the enteric nervous system to increase small intestine motility. The gastroileal is going to relax that sphincter, the ileocecal sphincter. And then gastrocolic increases movement along the colon. So now we're going to go through again, going over the secretions. What secretions are there, what function, and what regulates them. We're going to start out again in the mouth again, top to bottom. This time, though, we do add in the pancreas and the gallbladder/liver. So we have our three extrinsic salivary glands, the parotid, the sublingual, the submandibular. These are going to be the main ones for secreting saliva. There are a few buccal glands, but they mainly contribute some mucus. Saliva It's highlighted because you do need to know the components in saliva. Saliva is mostly water. It is slightly acidic. We produce quite a bit, a liter and a half per day. It does have additional substances such as defensins and lysozyme to prevent bacterial growth, as well as IgA antibodies. Its purposes are to moisten food to help to form a bolus, and it contains salivary amylase, which will start the digestion of carbohydrates. There is very little absorption that goes on in the mouth other than as far as nutrients. Things like drugs and alcohol can be absorbed, but what we would consider like protein, so amino acids and monosaccharides and like fatty acids, that absorption is going to occur in the small intestine. Lubrication via the mucus and the glycoprotein mucin, which becomes sticky when exposed to water. Okay, so we have mucus, we have mucin, we have water, which is acidic water, defensins, some antibodies, and lysozyme. Control of salivation, that is that cephalic phase of the digestive reflex, I guess you call it. So the cephalic phase is mainly controlled by the parasympathetic system. This is going to be the vagus nerve. It starts with thought, smell, taste of food. And then there are also chemoreceptors that will monitor pH, and then mechanoreceptors. It can be inhibited by the sympathetic nervous system. So the sympathetic nervous system pretty much undoes what this parasympathetic is doing. And so that's what the sympathetic nervous system will cause dry mouth, because it's saying you don't need to worry about eating and digesting right now. So normally control salivation is under parasympathetic nervous system activity, as well as that cephalic part of the gastric motility reflex. The esophagus doesn't secrete much. There is some mucus. We're going to go here. So for the mouth, the secretions are saliva, know those components, as well as things like defensins, lysozyme, and then salivary amylase. For the pharynx and the esophagus, not too much of a contribution. Mucus is the primary one. Then we get to the stomach. There are going to be a lot. We have gastric juice, hydrochloric acid, which is going to be secreted by the parietal cells. So make sure you can match the cell type of the gastric gland to its secretion. Pepsin, which, so I'm going to put pepsin, but it's really maybe I'll put, okay, I'll put both, just so you remember. So the teeth cells are going to secrete pepsinogen, which then gets clipped and activated further up by the hydrochloric acid will help with that. All of our protein digestive enzymes, whether they're secreted by the stomach or by the pancreas, are going to be secreted in a pro form. And that's because if they were secreted in an active form, then we would be digesting those glands and digesting those organs. So sometimes I'll write the active form like pepsin, but just know that I mean pepsinogen, because probably you're not going to want to write it both ways. Then we have mucus from the neck cells and a whole bunch of hormones. So the hormones that the stomach is going to release are gastrin, which is going to increase gastric juice. And then we're going to have histamine, which will increase hydrochloric acid, and somatostatin, which in the presence of acid inhibits everything. Even though I pre-wrote it on the other slide, stomach secretions are, we're going to get mucus throughout the stomach. It's actually a bilayer of mucus. So there's a layer of mucus, then in between, like the jelly, is a bicarbonate solution, and then another layer of mucus. So you've got this double layer with bicarbonate in between to protect the stomach. Those then we have the chief cells that are going to produce, and also remember where these things are located. Okay, so we have our enteroendocrine cells here at the bottom, and then we have our chief cells and then further up we have our parietal cells, and then we have our mucous neck cells. So also remember where these cells are located. So we have that double layer of mucus. We have chief cells that are producing pepsinogen, which will then get converted into pepsin. We have our parietal cells secreting hydrochloric acid. We have additional cells that are producing intrinsic factor, which we'll talk about more on the absorption digestion lecture, and gastrin. So then we have the hormones. Okay, so these are secretions, and then we have hormones. Gastrin is produced to, it will increase what's called gastric juice production. Histamine is going to increase hydrochloric production. So both gastrin and histamine are going to promote motility and digestion. The other hormone that we have is somatostatin, and it is going to inhibit gastrin and histamine. So for secretions, we have mucus through those neck cells, pepsinogen through the chief cells, hydrochloric acid through the parietal cells, as well as intrinsic factor. The hormones that are produced by the stomach are going to be gastrin and histamine. Both of these are going to promote motility and digestion. Gastrin through gastric juices, histamine through hydrochloric, but just know them collectively as enzymes, as hormones that promote motility and digestion. Then we have the hormone somatostatin, which inhibits stomach motility and digestion. This is just a reminder of where these cells are located. So we have the enteroendocrine cells. These are the cells here at the bottom that are going to produce gastrin, histamine. We didn't really go into serotonin and somatostatin. So these two promote stomach motility and digestion. Somatostatin inhibits stomach motility and digestion. And these can be found at the bottom of the gastric gland. If you move up the gastric gland, then you have the chief cell, which is secreting pepsinogen, which will become pepsin to digest proteins. But again, any protein digestive enzyme is going to be secreted in a pro form because we don't activate it until we get into the lumen where it can interact on the proper proteins. Then if you move up a little bit more, we get the parietal cells producing hydrochloric acid and intrinsic factor. That HCl environment is going to help break down proteins, but it'll also help to activate that pepsinogen into pepsin. And then last but not least, we get the mucus neck cells at the very top, producing an acidic version of mucus. So know the cells, know their secretions, and know these three hormones as far as do they promote stomach motility and digestion or inhibit. Okay, so then we have the small intestine secretions. Most of the secretions that act in the small intestine are produced either by the pancreas or the gallbladder. Okay, so the pancreas and the gallbladder will secrete enzymes and substances into the duodenum. The small intestine does secrete its own slightly basic water for motility, as well as secretin and cholecystokinin, which we'll get to. So for the pancreas is going to secrete, let's go back here. So we can go to the small intestine. The small intestine does secrete mucus and a slightly alkaline water for fluidity. It is going to secrete cholecystokinin, which increases bile and pancreatic enzymes, which we are just about to get to. It also secretes secretin, which increases bicarbonate from the pancreas. And then I'm going to put these down now so that they're all on the same table, but we're not going to talk about them really until we get to absorption and digestion. So then there are brush border enzymes. These are on the microvilli, and they are the last stage for breaking things down. We'll have disaccharidases, which are going to break down disaccharides to monosaccharides. Again, we'll go over the brush border enzymes a lot more in our next lecture, but this way you have them all in the same place, as well as aminopeptidases. And there is a bunch of these, chymotrypsin, trypsin, we'll go into those, that break polypeptides to amino acids. For the large intestine, we'll have mucus, we'll have vitamin K that is produced by bacteria, and then also some bicarbonate. For the pancreas, I'm actually, I might need to use both of these boxes. So both of these boxes are going to be secretions, but there's quite a bit. We have the duct cells that produce bicarbonate. And then the acinar cells are going to produce pancreatic lipase, amylase, and nuclease. So make sure I get them all in. Carboxypeptidase, trypsin, and chymotrypsin. Okay. The gallbladder is going to produce bile, which are bile salts, lecithin, cholesterol, bilirubin, and bicarbonate. And so that's it for this table. So I'm going to give you guys a minute to
So for gallbladder, I noticed there was nothing in the motility box. Is that?
You do for the gallbladder, as far as motility, you will need to understand the duct system, but that's technically part of the anatomy. So you do need to know like the cystic duct, and that is formed from the right and left going to the common hepatic, and then those join to make the common bile duct. So that diagram, there's actually 2 slides of that diagram. You'll have something that says like, here's a purple star, here's a yellow circle, you know, what duct is this? But nothing as far as actual, I guess, motility processes.
Okay, gotcha. Thank you.
Okay. So I'm going to go back. I can, at the end, I can come back to. Okay, so for the pancreas, we have duct cells and acinar cells. And the duct cells are responsible for bicarbonate production. So bicarb is going to neutralize the acidic chyme, and it is under hormonal control from secretin, which is produced by the small intestine. So if the small intestine has chemoreceptors and it says, oh, you know, like this is way too acidic, it will tell the stomach to slow down, and then it also tells the pancreas to secrete more bicarb. The acinar cells, these are the ones that produce the digestive enzymes. So there are proteases. Those are going to be carboxypeptidase, trypsin, and chymotrypsin. We'll talk about more of that with digestion for that digest proteins, pancreatic lipase for fat digestion, pancreatic amylase for starch digestion. So this is in addition to salivary amylase. And even so, both of these are only going to get them down to disaccharide level. And then the brush border enzymes are responsible for going disaccharide to monosaccharide, but we'll go over that at the next lecture. And pancreatic nuclease for nucleic acid digestion. So the pancreas produces all of these enzymes, and then it secretes them at the hepatopancreatic ampulla into the duodenum for action. The pancreas also has endocrine functions. It secretes insulin and glucagon. Those will not be on your test. Those are endocrine functions. Okay, so now we're to the liver and the gallbladder. Bile is produced continuously by the hepatocytes in the liver. And then it flows through the bile canaliculi, and it will go then through those right and left hepatic ducts to the common hepatic duct to the cystic duct. And that cystic duct is bidirectional. So bile can go up and get stored in the gallbladder, or it can go back down the cystic duct into the common bile duct for secretion at the duodenum. So bile contains bile salts and lecithin. Both of these are going to be important in emulsifying fats. Fats left to their own devices will form globules, and then it's very hard to digest them because it's a big globule of oil or fat. So the bile salts and the lecithin are going to be and very important in breaking those into much, much smaller droplets, which are easier to digest, accessed by the lipases. Bile also contains cholesterol, bilirubin, which is a breakdown product, as well as some bicarbonate. The two major ingredients in bile are bile salts to emulsify those fats. Bile release is stimulated by cholecystokinin, which is released by small intestine, and then bicarbonate, which is stimulated by secretin. So these two, CCK and secretin, these are small intestine hormones that are released, and they affect, they're going to promote digestion. Back to the small intestine. The small intestine does have a mucus lining to protect it from proteases and provide for lubrication. They have what are called crypts that secrete this water electrolyte solution. And then the brush border enzymes we'll get more in depth into in the next lecture. They're very interesting, but they also make sense as far as their function. So that's really nice, but they are a very elegant little system. This is reviewing the hormones of the small intestine. So the small intestine does secrete secretin and cholecystokinin. Both of these are going to aid in digestion. Secretin stimulates bicarb, cholecystokinin it stimulates the pancreas and the gallbladder. So for these two, you just need to know that they are secreted by the small intestine. Okay, so they're both secreted by the small intestine, and they both are going to promote or assist in digestion. Okay, you won't need to know all the intricate details of both of these. Large intestine, bicarbonate and mucus. And then that is it. We are right on time for those two trips. So I'm going to go back to these just in case you need, and if you need me to go up to the there you go. If you need me to go up a slide, then let me know and I can go up a slide, but I do want to make sure that you have and you know, eventually I will post this lecture. I'll be out of town again for the weekend for Parents' Day weekend, but I'm going to try and get these posted soon so that you at least have access to them. So for those of you who are good, I will see you after the weekend. If you have questions, if you want me to go back a slide, this is the time that you get to tell me what to do.